Photosynthesis NCERT based single sentence questions. Let's start. Physicochemical process which occur in green plants we call photosynthesis. Variegated leaf experiment explains photosynthesis occurred only in the green parts of the leaves in the presence of light. Light energy into organic compound, the process called physicochemical process. Test tube containing some KOH soaked cotton half leaf inside. Experiment for carbon dioxide was required for photosynthesis. Revealed the essential role of air in the growth of green plants. Joseph Priestley. Primary source of all food on earth. Photosynthesis. Responsible for the release of oxygen into the atmosphere. Photosynthesis. Performed a series of experiments in 1770. Joseph Priestley. Placing bell jar once in the dark and once in the sunlight. John Ingenhouse. Split spectral light, more bacteria on cladophora. Blue and red light. Split spectral light promote photosynthesis. Blue and red light. Discovered oxygen in 1774. Joseph Priestley. Light energy, carbon dioxide, water can make carbohydrates. Carbon dioxide dissolves in the water and in the presence of light, what it can make? It can make CH2O plus oxygen. 2H2A plus CO2 in the presence of light can make 2A plus CH2O plus H2O. Six CO2 plus 12 H2O in the presence of light can make C6H12O6 C6H12O6 plus 6H2O 6H2O plus 6O2. Aquatic plant bubbles from green plant in the presence of light. CH2O represent carbohydrates. Aquatic plant no bubbles from the green plant in the absence of light. Aquatic plant bubbles from green plant, bubbles are oxygen. Burning candle or animal that breathe the air experiment and scientist. Experiment, bell jar experiment. Bell jar experiment and scientist name Joseph Priestley. Glucose is usually stored as starch. Bell jar, mouse stay alive, candle burn continuously when we place mint plant. Scientists proved the importance of oxygen. Joseph Priestley. Scientists provided evidence for the production of glucose when plants grow. Julius van Sack. Scientists proved the importance of light. Scientists who proved the importance of light. John Ingenhouse. Scientists, green substance in plant is located in special bodies within the plant cell. Julius von Sack. Scientists, only the green part of the plant that could release oxygen. John Ingenhouse. Scientists who demonstrated that photosynthesis is essentially a light dependent reaction. Cornelius van Niel. Scientists, the green parts in plant glucose is made usually stored as starch. Julius von Sachs. Scientists, the first action of spectrum of photosynthesis was thus described. T.W. Engelmann. Scientists who demonstrated hydrogen from a suitable oxidizable compound reduces carbon dioxide to carbohydrate. Cornelius van Niel. Scientists proved the importance of light with aquatic plant. John Ingenhouse. Scientists inferred that the oxygen evolved by the green plant comes from water, not from carbon dioxide. Cornelius van Niel. Scientists prism algae cladophora aerobic bacteria. T.W. Engelmann. Microbiologist, a milestone contribution to the understanding of photosynthesis. Cornelius van Niel. Scientists studied on cladophora and aerobic bacteria. T.W. Engelmann. Scientists studies on purple and green bacteria. Cornelius van Niel. Hydrogen donor in green plants. Water. Water in green plants oxidize to. Water in green plants oxidize to oxygen. Hydrogen donor in purple and green sulfur bacteria. H2S. Organisms which do not release oxygen during photosynthesis. Purple and green sulfur bacteria. Hydrogen donor in organisms who do not release oxygen during photosynthesis. H2S. Oxidation product is sulfur and sulfate depending on the organism and not oxygen in purple and green sulfur bacteria. Oxygen evolved by the green plant comes from water, not from carbon dioxide. This was later proved by using radioisotopic techniques. The correct equation that would represent the overall process of photosynthesis is therefore. The correct equation that would represent the overall process of photosynthesis is therefore the 6 CO2 plus 12 H2O in the presence of light, glucose is formed along with the water molecule. Along with the hydrogen molecule. Process in which light energy is converted into chemical energy in organic compounds is called physico-chemical process. For example, that we have photosynthesis as an example here. An experimental setup to show that carbon dioxide is essential for photosynthesis is Mole's half-leaf experiment. First of all, Who demonstrated that sunlight is essential for photosynthesis in plant? John Ingenhouse. Production of glucose takes place in green part of the plant is established by J.V. Sachs. The first action spectrum by photosynthesis was described by T.W. Engelmann. Name the algae which was used to describe action spectrum of photosynthesis. Cladophora. Who demonstrated that photosynthesis is light dependent reaction in which hydrogen form a suitable oxidizable compound reduces carbon dioxide to carbohydrate like 2HTA plus CO2. In the presence will find 2A, CH2O and water. Also concluded oxygen evolved during photosynthesis comes from water, not from carbon dioxide. The scientist is C.V. Nile. Photosynthesis does not take place in the green leaves of the plant, but it does so also in other green parts of the plant. True or false? True. Can you name some other parts where you think photosynthesis may occur? Mesophyll cells. Can you name some other parts where you think photosynthesis may occur? In modified roots and stems. Cells of leaves which have large number of chloroplasts. Mesophyll cells. Usually the dash align themselves along with the usually dash will align chloroplast align themselves along with the walls of the mesophyll cells walls of the mesophyll cell such that they get the optimum quantity of the incident light. Membranous system of chloroplast consists grana, stroma, grana, stroma lamellae and matrix of the stroma. Division of labor in chloroplast, present or absent? Present. System of chloroplast responsible for tapping the light energy and also for the synthesis of ATP and NADPH. Membranous system. Enzymatic reaction synthesize sugar which in turn forms starch in stroma. Light driven reactions of photosynthesis are called light driven reactions of photosynthesis are called photochemical reactions or light reactions. Reactions which are not the light dependent but dependent on light reactions are carbon reactions or we can say dark reactions. Separation of leaf pigments of any green plant is possible through paper chromatography. A dash separation of the leaf pigment shows that the color that we see in leaves is not due to a single pigment but due to dash pigments. A chromatographic separation of the leaf pigment shows that the color that we see in leaves is not due to a single pigment but due to four pigments. The pigment with bright or blue green in the chromatogram, chlorophyll A. Pigment yellow green, chlorophyll B. Pigment yellow, xanthophylls. Pigment yellow to yellow orange, carotenoids. In chloroplast, each packet of grana is called thylakoid. Series of thylakoids in chloroplast are called grana. Two grana are connected each other by stroma lamella. Photosystems present on thylakoid are PS1 and PS2. Light reaction photosynthesis occurs at thylakoids. Dark reaction occurs at stroma. Participants in non-cyclic photophosphorylation. Participants in non-cyclic photophosphorylation PS1 and PS2. PS1 and 2. Participants in cyclic photophosphorylation. Only PS1. Phosphorylation process which is considered as jet scheme, non-cyclic. Substances that have an ability to absorb light at specific wavelengths are pigments. Can you guess which is the most abundant plant pigment in the world? Chlorophyll. From the figure, can you determine the wavelength or color of the light at which chlorophyll show maximum absorption? Which chlorophyll A shows the maximum absorption? Blue. Does it show another absorption peak at any other wavelengths too? If yes, which one? Red. So given figure 13.3b, explain. Showing the wavelength at which maximum photosynthesis occur. Photosynthetically active radiation, the wavelength is 400 to 700 nm. Chlorophyll wavelengths are peak at blue and red. If action spectrum and absorption spectrum are overlapped, the rate of photosynthesis mainly matches with chlorophyll A. Is it true that the wavelength at which there is a maximum absorption by chlorophyll A, that is in the blue and the red region, also show higher rate of photosynthesis? True. How we conclude that chlorophyll A is the chief pigment associated with photosynthesis? Action spectrum, rate of photosynthesis, and the action spectrum, rate of photosynthesis matches with absorption spectrum. By looking figure 13.3c, can you say that there is a complete one-to-one overlap between the absorption spectrum of the chlorophyll A and the action spectrum of photosynthesis? Yes. Most of photosynthesis takes place at which wavelengths of visible spectrum? Blue and red regions. Some photosynthesis does takes place at the other wavelengths of the visible spectrum. True or false? True. Major photopigment which is responsible for trapping of light? Chlorophyll A. Accessory photopigments? Chlorophyll B and xanthophyll, carotenoids. Accessory photopigment transfer the absorbed light energy to chlorophyll A. Do accessory photopigment absorb light? Yes. Wider range of wavelength of incoming light to be utilized for photosynthesis is possible by accessory photopigments. Who protect chlorophyll A from photooxidation? Accessory photopigments. Light reactions at the photochemical phase include light absorption, water splitting, and oxygen release. Then formation of ATP and NADPH. Several protein complexes which are involved in light reaction are organized into two discrete photochemical dash within the photosystem 1 and photosystem 2. Light harvesting complexes, LHC. Photosystem 1 and 2 are named in sequence based on their discovery. LHC are made up of hundreds of pigment molecules bound to proteins. Photopigments are named in the sequence of their discovery and not in the sequence in which they function during the light reaction. True. Each photosystem has all the pigments except one molecule of chlorophyll A. Each photosystem has all the pigments forming a light harvesting system, also called antennae. Dash help to make photosynthesis more efficient by absorbing different wavelengths of light. Pigments of light harvesting system. Reaction center is formed by chlorophyll A molecule. Reaction center in both the photosystem is different. In PS1, the reaction center chlorophyll A has an absorption peak at 700 nm. Then hence is called P700. In the PS2, the reaction center chlorophyll A has an absorption peak at 680 nm. Hence, it is called P680. In which photosystem does the reaction center chlorophyll A absorb 680 nm wavelength of red light, causing electrons to become excited and jump into an orbit further from the atomic nucleus? Photosystem 2. In which photosystem electrons in the reaction center are excited when they receive red light of the wavelength 700 nm and are transferred to another acceptor molecule that has a greater redox potential? Photosystem 1. Excited electrons of photosystem 2 are picked by electron acceptor. Picked by electron acceptor, which passes them to an electron transport system consisting of cytochromes. Excited electrons of photosystem 2 are picked by electron acceptor, which passes them to an electron transport system consisting of cytochromes. Splitting of water is associated with photosystem 1 or 2? Splitting of water is associated with photosystem 2. The movement of electrons in photosystem 2 is considered as downhill in terms of an oxidation, reduction or redox potential scale. Electrons of photosystem 2 are not used up as they pass through the electron transport chain, but are passed on to the pigments of photosystem 1. Electrons are moved downhill again, this time to a molecule of energy rich NADP plus in photosystem 1. Water splitting complex is associated with photosystem 2. Addition of electron reduces NADP plus 2. Addition of electron reduces the NADP plus 2, NADPH, NADPH plus H plus. The whole scheme of transfer of electron starting from the PS2 uphill to the acceptor, down the electron transport chain to PS1, excitation of electron, transfer to another acceptor, and finally downhill to NADP plus, reducing it to NADPH plus. H plus is called the jet scheme due to its characteristic shape. This shape is formed when all the carriers are placed in a sequence and a redox potential scale. Jet shape. How does PS2 supply electron continuously? The electrons that were moved from photosystem 2 must be replaced. This is achieved by electrons available due to splitting of water. Water is split into two hydrogen ions and an oxygen ion, electrons and electrons. This create oxygen, one of the net products of photosynthesis, splitting of water. The electrons needed to replace those removed from photosynthesis 1 are provided by photosystem 2. H2O, splitting of water into 4H plus oxygen plus 4 electrons. Water splitting complex is physically located at on the inner side of the membrane of thylakoid. Living organisms have the capability of extracting energy from oxidizable substances and store this in the form of bond energy. Special substances which carry energy in their chemical bonds are ATP. The process through which ATP is synthesized by cells is named as phosphorylation. Synthesis of ATP from ADP and inorganic phosphate. Synthesis of ATP from ADP and inorganic phosphate in the presence of light is called photophosphorylation. When only PS1 is functional, the electron is circulated within the photosystem and the photophosphorylation occurs due to the cyclic flow of electrons. The process is called cyclic photophosphorylation. When the two photosystems work in a series, first photosystem PS2 and then the PS1, a process is called non-cyclic photophosphorylation. Non-cyclic photophosphorylation. A possible location where this could be happening is in the stroma lamella. Possible location, that is cyclic photophosphorylation. Possible location where this could be happening is in the membrane or lamellae of grana. Non-cyclic photophosphorylation. Stroma lamellae membrane lack PS2 as well as NADP reductase enzyme. Hence, it performs only cyclic photophosphorylation. The excited electron does not pass on to the NADP plus but is cycled back to the PS1 complex through the electron transport chain, that is cyclic photophosphorylation. Synthesize not only ATP and also NADPH plus H plus, that is non-cyclic photophosphorylation. Non-cyclic photophosphorylation. Only in the synthesis of ATP, that is cyclic photophosphorylation, occurs when only light of wavelengths beyond 680 nm are available for excitation. Cyclic photophosphorylation. Electron is circulating. The electron is circulated within the photosystem and the phosphorylation occurs due to cyclic flow of electrons when only PS1 is functional. Cytoorganelles which participate in photophosphorylation process are mitochondria and chloroplast. The two photosystems are connected through an electron transport chain. Electron transport chain of the Z scheme. Both ATP and NADP H plus are synthesized by this kind of electron flow. Electron transport chain form from PS2 to PS1. PS2 to PS1, that is a Z scheme. When only PS1 is functional, electron is circulated within the photosystem. When only PS1 functional, phosphorylation occurs due to cyclic flow of electrons. Hypothesis which explains how actually ATP is synthesized in the chloroplast. Chemiosmotic hypothesis. Like in respiration, in photosynthesis too, ATP synthesis is linked to the development of a proton gradient across the membrane. ATP synthesis is linked membranes of thylakoid. Linked with the membranes of thylakoid. If proton accumulation is towards the inside of the membrane, that is lumen, it is Photosynthesis. If proton accumulate in the intermembrane space of the mitochondria, when electron move through the electron transport, it is respiration. What causes the proton gradient across the membrane? By splitting of water, by plastoquinone, or by NADP reductase enzyme? All the three. Splitting of the water molecule takes place on the inner side of the membrane. The protons are hydrogen ions that are produced by the splitting of water accumulate within the lumen of thylakoids. Electron move through photosystems. Protons are electron move through photosystem. The protons are transported across the membrane. This happens because the primary acceptor of the electron, which is located towards the outer side, of the membrane, transfers its electron not to an electron carrier but to an hydrogen carrier. Hence, this molecule removes a proton from the stroma, while transporting an electron. When this molecule passes on its electron to the electron carrier on the inner side of the membrane, proton is released into the inner side or lumen side of the membrane. The NADP reductase enzyme located on the stroma side of the membrane along with electrons that come from the acceptor of electron of PS1. Protons are necessary for the reduction of NADP plus to NADPH plus and H plus. These protons are also removed from the stroma. Hence, within the chloroplast, protons in the stroma decrease in number, while in the lumen of thylakoid, in the lumen of thylakoid, there is an accumulation of the proteins, lumen of thylakoid. This creates a proton gradient across the thylakoid membrane as well as a measurable decrease in pH in the lumen. That is by splitting of water. There are three ways: by splitting of water and by plastoquinone and by NADP reductase enzyme. By splitting of water, by plastoquinone while carrying the electron, then by NADP reductase while converting NADPH process, it also transport a proton of hydrogen atom into the lumen of thylakoid. Why are we so interested in the proton gradient? This gradient is important because the breakdown of this gradient leads to the synthesis of ATP. Accumulation of protons from stroma of chloroplast, to lumen of thylakoid by three ways: by splitting of water, by plastoquinone, by NADP reductase enzyme. The concentration gradient of protons more at lumen of thylakoid. Movement of protons due to the concentration gradient from stroma of chloroplast to lumen of thylakoid. What is CF0 and CF1 in photosynthesis? Proton channels. The concentration gradient of protons in the lumen of thylakoid is broken down due to the movement of proton across the membrane to the stroma through the transmembrane channel of CF naught of the ATP synthase. Two parts of the ATP synthase enzyme are the two parts of ATP synthase enzyme are CF naught and CF1. Part of ATP synthase enzyme, which is embedded in the thylakoid membrane and forms a transmembrane channel that carries out facilitated diffusion of protons across the membrane, are CF0. Part of ATP synthase which protrudes on the outer surface of the thylakoid membrane, one at the side that faces the stroma, that is CF1. Breakdown of the gradient provides enough energy to cause conformational change in the CF1 particle, conformational change in the CF1 particle of the ATP synthase, which makes the enzyme synthesize several molecules of energy packed ATP. Chemiosmosis require a membrane and a proton pump and a proton gradient and ATP synthase enzyme. ATP synthase enzyme. Energy used to pump protons across a membrane to create gradient or a high concentration of protons within the thylakoid lumen. Dash has a channel that allows diffusion of protons back across the membrane, and this releases enough energy to activate ATP synthase enzyme. And Dash has a channel, ATP synthase has a channel that allows diffusion of proton back across the membrane, and this release enough energy to activate ATP synthase enzyme that catalyzes the formation of ATP. Along with the NADPH produced by the movement of electron, the ATP will be used immediately in the biosynthetic reaction taking place in the stroma, that stroma responsible for the fixing of carbon dioxide and the synthesis of sugars. We need to emphasize here that water splitting complex is associated with the PS2, which itself is physically located on the inner side of the membrane of the thylakoid. Then, where are the protons and oxygen formed likely to be released in the lumen or in the on the outer side of the membrane? The oxygen is released on the outer side of the membrane. Oxygen is released on the outer side of the membrane, whereas the protons are released from the lumen. Products of light reaction are ATP, NADPH, and oxygen. In the products of light reaction, who diffuses out of the chloroplast, that is oxygen. In the products of light reaction, ATP, NADPH are used to drive the process leading the synthesis of food, and this is the more accurately sugars. and this is the biosynthetic phase of photosynthesis. This process does not directly depend on the presence of light, but is dependent on the products of the light reaction, that is ATP, NADPH, besides carbon dioxide and water. That is biosynthetic phase of photosynthesis. Biosynthetic phase of photosynthesis. Immediately after light becomes unavailable, the dash continues. Biosynthetic phase continues for some time and then stops. If the light is made available, the biosynthetic process starts again. Can we hence say that calling the biosynthetic phase as a dark reaction is a misnomer? Yes, because they do not require darkness. They do not require darkness, they are merely light independent. They are merely light independent. That means dark reactions occur during the day. Dark reactions occurs during the day because they are a part of photosynthesis. Just after World War II, among the several efforts to put radioisotopes to beneficial use, the work of this scientist is exemplary. Melvin Calvin. Use of radioactive C14 by him in algal photosynthesis studies led to the discovery that the first carbon dioxide fixation product was 3-carbon organic acid, that is Melvin Calvin. He also contributed to working out the complete biosynthetic pathway. Hence, it was called the cycle after him. Melvin Calvin, hence it was called Calvin cycle after him. The first product identified in C3 cycle was 3-phosphoglyceric acid, or in short, PGA. True. 3-phosphoglyceric acid, or in short, it is called PGA. Experiments conducted over a wide range of plants led to the discovery of another group of plants where the first stable product of carbon dioxide fixation was again an organic acid, one which had 4 carbon atoms in it. This acid was identified to be oxaloacetic acid, OAA. Carbon dioxide assimilation during photosynthesis was said to be of two main types. Those plants in which the first product of carbon dioxide fixation is C3 acid, that is PGA, phosphoglyceric acid, and the oxaloacetic acid, OAA, and those in which first product was C4 acid. Carbon dioxide assimilation during photosynthesis was said to be of two main types. Those planned in the first product of CO2 fixation is C3 acid, that is PGA. Hence, cycle called C3 cycle. And those in which the first product was C4 acid, that is oxaloacetic acid, that is C4 cycle. In C3 plants, cell type in which the Calvin cycle takes place, that is mesophyll cells. In C3 plant, cell type in which the initial carboxylation reaction occurs, mesophyll cells. In C3 plant, how many cell type does the leaf have that fix carbon dioxide? One, that is one mesophyll. In C3 plant, which is the primary carbon dioxide acceptor, RUBP. In C3 plant, number of carbons in the primary carbon dioxide acceptor. 5. In C3 plant, which is the primary carbon dioxide fixation product, phosphoglyceric acid. In C3 plant, number of carbons in the primary carbon dioxide fixation product. 3. Does the plant have Rubisco? Yes, no, not always. In C3 plant, does the plant have Rubisco? Yes. In C3 plants, does the plant have pepcase, PEP? No. In C3 plant, which cells in the plant have Rubisco, mesophyll, bundle sheath or none? That is in mesophyll cells. In C3 plant, carbon dioxide fixation rate under light conditions, high, low or high, medium? Low. In C3 plants, whether photorespiration is at low light intensities, negligible. Whether photorespiration is present at high light intensities, high, negligible, sometimes, high. In C3 plant, whether photorespiration would be present at low carbon dioxide concentration is high, negligible, or sometimes, high. In C3 plant, whether photorespiration would be present at high carbon dioxide concentration is high, negligible, or sometimes, negligible. In C3 plants, temperature optimum 30 to 40 degrees centigrade or 20 to 25 degrees centigrade, above 40, 20 to 25 degrees centigrade. In C3 plant, examples like the cut vertical sections of the leaves of different plants and observe under the microscope for cranch anatomy and list them in appropriate column. That in C3 plants column, we get wheat and rice. Let's go with C4 plants. C4 plant, cell type in which Calvin cycle takes place, mesophyll or bundle sheath or both. C4 plants, that is bundle sheath. Cell type in which the initial carboxylation reaction occurs, in mesophyll. In C4 plants, cell type in which the initial carboxylation reaction occur, mesophyll. In C4 plants, how many cell types does the leaf have that fix carbon dioxide? 2. And the option 2, that is bundle sheath and mesophyll. In C4 plant, which is the primary carbon dioxide acceptor from the given options? PEP. In C4 plants, number of carbons in the primary carbon dioxide acceptor? 3. In C4 plant, which is the primary carbon dioxide fixation product? OAA, oxaloacetic acid. Number of carbons in the primary carbon dioxide fixation product? 4. Does the plant have Rubisco? Yes. In C4 plants, does the plant have PEP case? Yes, no, not always. In C4 plant, does the plant have PEP case? Yes. In C4 plants, which cells the plant have Rubisco? Bundle sheath. Carbon dioxide fixation rate under light conditions in C4 plants? High. Whether photorespiration is at low light intensities, high, negligible, or sometimes present? Negligible. Whether photorespiration is present at high light intensities, high, negligible, or sometimes present? Negligible. In C4 plants, whether photorespiration would be present at low carbon dioxide concentration? Negligible. In C4 plants, whether the photorespiration would be present at high carbon dioxide concentrations. Negligible. Temperature optimum in C4 plants 30 degrees to 40 degrees. In C4 plants, example, cut vertical sections of the leaves of different plants and observe under the microscope for cranch anatomy and list them in appropriate column. Those who have cranch anatomy, they are C4 plants, maize, sugarcane, and sorghum. The rate of photosynthesis is very important in determining the yield of plants, including crop plants. Photosynthesis is under the influence of factors like both internal plants or external plants, both external and internal plants. The factors include the number, size, age, and orientation of leaves, mesophyll cells, and chloroplast, and internal carbon dioxide concentration, and the amount of chlorophyll. Internal factors or external factors are both. Internal are plant factors. Internal factor, nothing but plant factor. The factors which are dependent on the genetic predisposition and the growth of the plant. Internal factor or external factor. Internal are plant factors, you can see. External factors should include the availability of sunlight, temperature, carbon dioxide concentration, and water. Internal factor or external factor. These are external factors. As a plant photosynthesis, all these factors will simultaneously affect its rate, internal or external or both factors. When several factors affect any biochemical process, the dash law of limiting factor comes into effect. Blackman's limiting factor. Hence, though several factors interact and simultaneously affect photosynthesis or carbon dioxide fixation, usually one factor is the major cause or is the one that limits the rate. Hence, at any point, the rate will be determined by the factor available at suboptimal levels. True or false? True. This states the following: if a chemical process is affected by more than one factor, then its rate will be determined by the factor which is nearest to its minimal value. It is the factor which directly affects the process if its quantity is changed. That is Blackman's law of limiting factor. For example, despite the presence of a green leaf and optimal light and carbon dioxide condition, the plant may not photosynthesize if the temperature is very low. This leaf, if the given the optimal temperature, will start photosynthesize. The given example explains Blackman's law of limiting factor. We need to distinguish between light quality and light intensity while discussing the light as a factor that affects photosynthesis. We have to cover exposure to light. There is a dash relationship between the incident solar light and carbon dioxide fixation rate at low light intensities. Linear relationship. At higher light intensities, gradually the rate does not show further increase as other factors become limiting factors. Except for plants in shade or in dense forests, light is rarely a limiting factor in nature. Why? Why? Light saturation, because light saturation occurs at 10% of the 10% of the full sunlight. Effect of increase in incident light beyond a point on chlorophyll and photosynthesis causes the breakdown of chlorophyll and a decrease in photosynthesis. Causes the breakdown of chlorophyll and a decrease in photosynthesis. Major limiting factor for photosynthesis, carbon dioxide. Major limiting factor for photosynthesis is carbon dioxide. Though the light reactions are also temperature sensitive, they are affected to a much lesser extent. True or false? True. Concentration of carbon dioxide in atmosphere between 0. Between 0.03 and 0.04%. Increase in carbon dioxide concentration up to 0.05% can cause damage over long periods if it is present for long period. Response of C3 and C4 plant against carbon dioxide concentration is same or different? Different. Different. At high light intensities, which plant shows? Okay, here we have one more question. Increase in carbon dioxide concentration up to 0.05% can cause increase in carbon dioxide fixation rates. Increase in carbon dioxide concentration beyond up to 0.05% carbon dioxide, it affects an increase in carbon dioxide fixation rate, but more than beyond 0.05% damage over long periods. Response of C3 and C4 plants against carbon dioxide concentration is different. At high light intensities, which plants show increase in the rate of photosynthesis? Both. Saturation levels of carbon dioxide in C3 plants 450 microliters, 450 micron per liter. Saturation of levels of carbon dioxide in C4 plants, that is 360. Current availability of carbon dioxide levels is limiting to the C3 plants. The fact that dash plants respond to higher carbon dioxide concentration. C3 plants respond to higher carbon dioxide concentration by showing increased rates of photosynthesis leading to a higher productivity has been used for some greenhouse crops such as tomato and bell pepper. They are allowed to grow in carbon dioxide enriched atmosphere that lead to higher yields. That is C3. Reactions which are being enzymatic and temperature controlled are dark reactions. Temperature sensitive reactions are light reactions. The dash plants respond to higher temperatures. The dash plants respond to higher temperature. C4 plants respond to higher temperature and show higher rate of photosynthesis, while C3 plants have much lower temperature optimum. The temperature optimum for photosynthesis of different plants also depends on the habitat that they are adapted to. True. Tropical plants have lower temperature optimum than the plants adapted to temperate climates. False. Tropical plants have a higher temperature optimum than the plants adapted to temperate climates. True. Even though water is one of the reactants in the light reaction, the effect of water as a factor is more through its effect on the plant rather than the directly on photosynthesis. Water stress causes the stomata to close, hence reducing the carbon dioxide availability. Besides, water stress also makes leaves wilt, thus reducing the surface area of the leaves and their metabolic activity as well. The process which require chlorophyll, light and carbon dioxide. Photosynthesis. A sharp revision from here. Primary source of all food on earth. Photosynthesis. Responsible for the release of oxygen into the atmosphere. Photosynthesis. Basis of life on earth. Photosynthesis. Physicochemical process. Photosynthesis. Test for starch. Test for starch. Variegated leaf experiment. Occurs only in green parts of the leaves in presence of sunlight. Photosynthesis. Test for carbon dioxide. Half leaf experiment. Scientists to explain role of air in growth of green plants. Scientists to explain role of air in growth of green plants. Joseph Priestley. Scientists who discovered oxygen in 1774. Joseph Priestley. This is a sharp revision. Plant used in bell jar experiment. Plant used in bell jar experiment. Mint plant. Plants restore to the air whatever breathing animals and burning candles remove. Priestley's hypothesis. Scientist sunlight is essential for plant process. John Ingenhouse. Plant used in Ingenhouse experiment. Plant used in Ingenhouse experiment. Aquatic plant. Identified small bubbles to be of oxygen and showed that only green parts of plants release oxygen. Scientist Ingenhouse. Provide evidence for the production of glucose when plants grow. Julius Van Sachs. Usually stored as starch. Glucose. Green substances in plants is located in special bodies in plant cells. Scientist Julius Van Sachs. Eliminated a green algae cladophora placed in a suspension of aerobic bacteria. Scientist T.W. Engelmann. Used to detect sites of oxygen evolution. Bacteria in Engelmann experiment. Accumulated mostly in the region of blue and red light if split spectrum. Bacteria in Engelmann experiment. First action spectrum of photosynthesis. T.W. Engelmann. Carbon dioxide in the presence of water and light and form glucose along with the oxygen. The experiment empirical equation of photosynthesis. Scientists photosynthesis is essential and a light dependent reaction. Van Neal, 1897 to 1985. Hydrogen from a suitable oxidizable compound reduces carbon dioxide in carbohydrate, that is Van Neal. Scientists who made studies on purple and green bacteria. Van Neal. Hydrogen donor in green plants, water. Oxidized to oxygen, water. Hydrogen donor for purple and green sulfur bacteria, hydro H2S. Product in purple and green sulfur bacteria, sulfur, sulfur or sulfate. Evolved by green plants comes from water, oxygen. Equation, the given equation, photosynthesis. Occurs in green leaves of plants and green parts of plants. Photosynthesis. Contain large number of chloroplasts, mesophyll cells. Align themselves along the walls of mesophyll cells, chloroplast. Grana, stroma lamellae, fluid stroma, membranous system. Membranous system. Responsible for tapping light energy and also for synthesis of ATP and NADPH. Membranous system. Enzymatic reaction, sugar synthesis. Stroma. Membrane system is for light reactions. Stroma is for dark reactions. Separation of leaf pigments of any green plant can be on paper chromatography, can be identified on paper chromatography. Bright or blue-green pigment, chlorophyll A. Yellow-green pigment, chlorophyll B. Yellow pigment, xanthophyll. Yellow to yellow-orange pigment, carotenoids. Blue and red regions of Vibgea, higher rates of photosynthesis. Chief pigment associated with photosynthesis. Chief pigment associated with photosynthesis, that is chlorophyll A. Chlorophyll B, xanthophyll and carotenoids are called accessory pigments. Transfer energy to chlorophyll A, accessory pigments. Protect chlorophyll A from photooxidation, accessory pigments. Include light absorption, water splitting, oxygen release, and formation of high energy chemical intermediates ATP and NADP are light reactions or photochemical phase. Made up of hundreds of pigment molecule bound to protein, LHC, light harvesting complex. Antennae, light harvesting system, LHC, has all the pigments except molecules of chlorophyll. Photosystem forms reaction center, chlorophyll A. PS1 P700, absorption peak at 700 nm. PS2 P680, absorption peak at 680 nm. Consists of cytochromes, electron transport system. Energy rich, NADP plus. Scheme of transfer of electrons. Scheme of transfer of electrons, jet scheme. Splitting of water, PS2. One of net product of photosynthesis, oxygen. The equation, water splitting. Physically located on inner side of membrane of thylakoid. Water splitting complex. Synthesis of ATP by mitochondria and chloroplast. Photophosphorylation. Synthesis of ATP from ADP and inorganic phosphate in presence of light. Photophosphorylation. Two photosystems work in a series. Non-cycle photophosphorylation. Synthesis of both ATP and NADPH plus and H plus occurs in lamellae of grana. Non-cyclic photophosphorylation. Non-cyclic photophosphorylation occurs in stroma lamellae, lacks PS2 as well as NADP reductase enzyme. Synthesis of only ATP occurs when light of wavelength beyond 680 nm is available. That is cyclic photophosphorylation. ATP synthesis, chemiosmotic hypothesis. Development of proton gradient is for ATP synthesis. Proton accumulation occurs and needed at lumen of thylakoid. Protons accumulate in intermembrane space of mitochondria. Respiration. Located towards outer side of membrane. Primary acceptor of electron. Located on the stroma side of the membrane. NADP reductase. CF0 and CF1. CF0 and CF1. There where at ATPase enzyme is present. ATP synthase embedded in membrane and forms transmembrane channel. F0 particle, CF0 particle. Carries out facilitated diffusion of protons. Transmembrane channel. Protrudes on outer surface of thylakoid membrane on the side that faces stroma. F1 particle. Stroma responsible for fixing of carbon dioxide and synthesis of sugar. Dark reaction. Product of light reactions. ATP, NADPH and oxygen. Synthesis of sugars in biosynthetic phase. Synthesis, radioactive C14 in algal photosynthesis contributed to working out the complete biosynthetic pathways explained by Melvin Calvin. First carbon dioxide fixation product in C3 pathway, that is PGA, 3-phosphoglyceric acid. First carbon dioxide fixation in product C4 pathway, that OAA, oxaloacetic acid, occurs in all photosynthetic plants. Calvin pathway. Calvin pathway. Carboxylation, reduction and regeneration, C3 plants. Primary carbon dioxide acceptor in C3 pathway, Rubisco. Formation of glucose, reduction phase. Adapted to dry tropical region, C4 pathway. Special type of leaf anatomy, tolerate higher temperature, show response to high light intensities, lack photorespiration, greater productivity of biomass, that is in C4 plants. Vascular bundles of C4 pathway, bundle sheath cells. Frange anatomy seen in C4 plants. The Reth, frange. Several layers around vascular bundles, large number of chloroplasts, thick walls impervious to gaseous exchange. bundle sheath cells. Number of intercellular spaces. Maize, sorghum are examples for C4 plants. Hatch-Slack pathway is C4. Enzyme responsible for carbon dioxide fixation. PEP carboxylase enzyme. Lac rubisco enzyme in mesophyll cells. C4 plants. Formed in mesophyll cells oxaloacetic acid. C4 plants. Occurs in bundle sheath cells. Decarboxylation. Rich in rubisco. Lac PEP. Rich in rubisco. Lac PEP. That is bundle sheath cells. Common to C3 and C4 plants. Calvin pathway. So given equation explains photorespiration. Most abundant enzyme in the world has much greater affinity for carbon dioxide than oxygen. That is rubisco. So RUBP plus oxygen form phosphoglycerate and phosphoglycolate. That is photorespiration in C3 plants. Neither synthesis of sugars nor of ATP result in release of carbon dioxide with utilization of ATP. It's a wasteful process, does not occur in C4 plant as they have a mechanism that increases the concentration of carbon dioxide at enzyme site that is photorespiration. Explaining about photorespiration process. Number, size, age, orientation of the leaves, mesophyll cells, chloroplast, internal carbon dioxide concentration and amount of chlorophyll depends on genetic predisposition and growth of plant. That all internal factors affecting photosynthesis. Internal factors affecting photosynthesis. Availability of sunlight, temperature, carbon dioxide concentration and water. External factors affecting photosynthesis. Law of limiting factors is given by Blackman. Affects photosynthesis, quality, intensity, duration of exposure to light, affects rate of photosynthesis. Light. Linear relationship between incident light and carbon dioxide fixation at low light intensities. Rate does not show further increase at higher light intensities. Occurs at 10% of full sunlight, rarely a limiting factor in nature except for plants in shade or dense forests. That is saturation. Major limiting factor for photosynthesis concentration of carbon dioxide is vary in the atmosphere between 0.03 and 0.04%, that is about carbon dioxide. Do not respond to high carbon dioxide condition at low light intensities, that is C3 and C4 plants. Show saturation at about 360 micron per liter, that is in C4 plants. Respond to increased carbon dioxide concentration at saturation is seen only beyond 450, that is current availability of carbon dioxide level is limiting to the C3 plants. Greenhouse crops allowed to grow in carbon dioxide enriched atmosphere that leads to higher yields. Tomato and bell pepper. Temperature controlled enzymatic reactions. Dark reactions. Respond to higher temperature and show higher rate of photosynthesis. C4 plants. Have a much lower temperature optimum. C3 plants. Depends on habitat that the plants adapted to. Temperature optimum. Have a higher temperature optimum. Tropical plants. Have a lower temperature optimum. Temperate plants. Its effect more on plant rather than directly on photosynthesis. Water. Cause stomata close, hence reducing carbon dioxide availability, make leaves wilt, reduces surface area of leaves and their metabolic activity as well. Water stress. That's it. That is about photosynthesis. Thank you.